Hello, everyone. I'm Birgit Sawitzki, and I will be moderating today's BIH lecture, where we will have Jens Rittscher giving a talk on his favorite tools. And he's really an expert, and I'm delighted that he accepted to give this lecture on using AI and computational tools to analyze, study images, maybe cell-based, but also tissue sections. Jens has just recently accepted our invite to act as a member of the BIH Scientific Advisory Board. He's a professor of engineering sciences at the University of Oxford, so Institute of Biomedical Engineering and the Nuffield Department of Medicine. And he's also a group leader at the Big Data Institute there in Oxford, and is affiliated with the Ludwig Institute of Cancer Research and the Wellcome Center. And we also applied for him to become a Charité visiting fellow, which was successful. So you will have the chance, he is visiting Berlin regularly, you will have the chance to talk to him also after this lecture today, and might start some more closer collaboration, which we will definitely do. So without further adu, I would really ask Jens now to start the lecture. As you are familiar with the process, please post the questions in the chat, because you will not have the chance to speak out freely. And I will then try to raise the questions you posted in the chat, as long as we don't get over the time that much. So please post all the questions in the chat. Jens, the stage is yours, and I'm looking forward to the presentation and a lively discussion afterwards.
Okay, thank you. So I hope you can hear me well and see the slides. First of all, thank you, Birgit, for the introduction and the opportunity to speak. I have a couple of conflicts of interest to declare. So we have a spin-out company in Oxford, and some of the work is also commercially funded. What I really wanted to do is use this opportunity to to present a little bit what our group is doing. As Birgit mentioned, we really sort of work at the microscopic scale to look at cells and tissue, developing computer vision algorithms and AI methodologies to sort of analyze image data. We also sort of focus on the analysis of larger data sets, sort of clinical cohorts. I will illustrate that a little bit throughout the talk. And finally, we're interested in the translational aspect of these technologies as well. So I give an example at the end, where we now basically started the first clinical evaluation studies. The first piece of work I wanted to present is really around basically ultrastructure imaging. This is a close collaboration with Jörg Beversdorf in Yale. It stems out of a collaboration in basic science, which is funded by the Wellcome Trust for many years. So what Jörg really developed is an expansion protocol that then uses a pan staining methodology, where we can essentially get contrast throughout the cell. We can increase the cell size by a factor of 10 to 15. And what is really quite remarkable is that by applying NHSS as a staining, we can visualize the entire cell. And you can see here there are two little arrows. So Jörg is a physicist, and as you heard, I'm purely computational. So we thought a good first topic would be to analyze mitochondria and mitochondrial morphology. And this is something we have been doing now. And I give you some examples in fixed cells. We've worked in tissues as well. But the the benefit of this technology is that through basically expansion microscopy, you get images that are comparable, probably not as exquisite in quality with electron microscopy. However, the expansion protocol can be run on a normal confocal microscope, and therefore it can be done really with higher throughput and in more labs. The other advantage is that we can still use stains, molecular labels, what you see here, to sort of highlight areas, for instance here, the cristae structure in the outer membrane of the mitochondria, to first of all visualize specific structures we're interested in, but more importantly, we can also use that to basically train algorithms, segmentation algorithms. And this is largely what this piece of work is about. So here we have a workflow which consists out of pre-training. We're essentially using unit variants for the training, but here, for instance, it's the training is aided by the mitotracker to essentially find mitochondrial structures, and then the training is refined through a level of annotations. So here the whole workflow is we work on the NHS ester, we predict essentially where structures of interest are, have segmentation, and then use fine-tuning, and then we can extract all sorts of parameters, like we can do skeletonization, we can look at mean thickness, branching points, and store all these parameters for further analysis. So the first step, as I outlined, is really based on the input image to train a unit to predict the mitotracker segmentation mask. And then throughout basically manual annotation, we look to basically refine this so that we get the internal structure, so we get the cristae structure, and the membranes can segment these and also analyze these later on. One thing that was quite an important experiment in our work was to evaluate how much annotations are necessary to get essentially usable images. So what you see here on the right is you see a comparison, especially on the lower right, of sort of where we just annotate on single slices, not the entire 3D image, on 140 slices. And you see that actually the performance metrics in terms of a dye score are actually not that divergent. That means that, for instance, a new lab could take the base segmentation model, adapted with relatively little annotation to a new imaging problem. So here we've essentially shown that we don't really need to annotate every slice. So we can annotate just a selection, a subsection of slices, and really still get quite a lot of utility out of these annotations for segmentation. This is now a video that shows you basically going through the Z-stacks of the cells, how we see the different mitochondria. Here you now see the segmentation and also the cristae membrane structure. I have a similar video side by side, so you get a better idea of how complete the segmentation is. We are fully aware that if we now use a different cell type, these are basically HeLa cells, that the mitochondrial structure will probably look slightly different. I can comment on this a little bit at the end. We've also made experiments in tissues, and of course there some of the quality is not as exquisite as you've seen here. But we will have a full 3D volume, and out of this 3D volume we can now extract the whole mitochondrial network. And as you can also see, we can now basically look at the connected components of that network, and we can study this in different kinds of conditions. So for example, before I do this, here's just a Now, a set of very generic measurements we can extract. So you see, for instance, the percentage of cristae, the inter-cristae distances, the number of mitochondria in terms of average diameter and length. And then we also can analyze the branching points and the frequency of those. So as a first experiment, we basically treated HeLa cells with different drugs. So we used 4 drugs here. Three different drugs: HBSS, oligomycin, and antimycin. We selected those because there's a fair amount of knowledge about these drugs in the literature. And here I basically show you the comparison between control and oligomycin. The control is on the top row, the oligomycin is on the bottom row. And here what you see is now the quantitative comparison between the control and oligomycin. And you see that in terms of fraction of cristae volume, length, which is in panel E, and for average diameter, well, in average diameter, you don't see such a big difference, but in terms of length and average planar intercristae distances, you see some differences that actually match with the expectations from other previously published work. So I'll comment a little bit on this at the end, but essentially this is a technology that could be now applied to multiple different organelles, and for instance also studies in cells and tissue. I'd like the opportunity now to talk a little bit about our pathology work. And here we use AI almost like for looking at 3 different sort of core aspects. One is to improve the accuracy of quality of diagnosis. That's essentially supporting pathologists. And I give an example of this. We can also look for new ways to sort of look at prognosis and monitoring therapy. I'm going to illustrate that in the context of colorectal cancer. And overall, we're interested in also overcoming the limitations of categorical grading and look at, for instance, more refined measures. And here we'll illustrate our work on fibrosis scoring. So before I do this, we are in a fortunate situation in Oxford that the cellular pathology in our trust is completely digital, and it has been completely digital since the summer of 2020. That means we actually were able to use the digital platform throughout the pandemic. We digitize about 13 to 1500 slides per day, and more than 20 consultants are basically set up to report out digitally across all subdisciplines. And this is, UCAS accredited, which is a requirement in the UK. And we were lucky that Philips, through a collaboration, through the UK Life Science Initiatives, we had funding from Philips and the UK government to basically pay for all the digitization. One of the initial works we did is we tried to see how we could sort of relate morphology to molecular stratifiers. And here we had quite a lot of expertise because Tim Morn, he established the ESCORT cohort for the purpose of stratifying colorectal cancer patients. And he had quite a lot of experience of basically the CMS, the common molecular subtypes of colorectal cancer, that were so far routinely done through RNA sequencing. So we developed an image-based approach where we essentially look at morphology subtypes to predict those CMS classes. Now, this is a few years ago, but the question was also, can we use that in a context of also looking at response to therapy? And this is work that Ruby Wood has done throughout her PhD while she was in Oxford. And we know that there are a number of colorectal patients that don't really respond well to radiotherapy. So we basically thought, well, can we actually use our knowledge about CMS and knowledge about morphology to essentially predict which patients would probably have, are more likely to have a complete response to radiotherapy. And we know that, for instance, the spatial organization, the interaction between epithelium and other compartments are important and have prognostic relevance. We know that CMS patients typically don't have such a good response to radiotherapy. And we also know that the stromal invasion that's associated with CMS4 is an indication of worse survival. And we try to find a way of actually predicting a couple of things at the same time, and you're going to see in a minute how we do this. The initial data set we worked on is relatively small. It's part of the Aristotle trial, and we have essentially 320 patients with CMS4, 45 of them not CMS. We're now looking at almost like representing the image as a graph, and this is typically done in histology imaging. So this is nothing radically novel. So you see the input slide on the left, and first of all, we segment this in just sets of cells that have roughly similar morphology using a superpixel, which is a very well developed technology. We used then self-supervised features. These are essentially the Dino features, but that could be now replaced with any feature set from a foundation model. Then we construct a graph. And the novel piece of the work was really that we then sort of use a graph neural network to predict three different things at the same time. So we predict response to therapy. We predict essentially the presence of CMS4 at at the basically microscopic level, and then we also predict the epithelial boundaries, and we do this all at the same time, which actually has a number of advantages, and you're going to see that in a minute. This is just for those who are interested in the loss function we used. The basically AUC values of response to therapy and CMS4 are well over 80%, so they are quite acceptable. The epithelial boundaries could be improved, but that could also relates to the size of the superpixels. We had no, basically so far we haven't really spent a lot of time improving this. But what you see here is essentially the output of this algorithm. We have now almost like three different masks. We have a response to therapy, we have CMS4, and we have the epithelium mask. And Victor Koser, the collaborating pathologist, has actually gone through a number of cases and almost like narrated them. And we sort of see here, for instance, high CMS4 activation response to low activation in response to radiotherapy. And we also sort of see here that high CMS4 activation corresponds to stromal region, or is in stromal regions. So the idea is that, for instance, an oncologist could communicate with a pathologist in a level that goes beyond a short pathology report, and could sort of get some input that could, for instance, guide therapeutic decisions. The next set of work, which is far more advanced, is really the work we've done on myeloproliferative neoplasms. This is a type of hematological malignancy. And this work has largely been led by Daniel Royston, who is a consultant hematologist and also academic pathologist in Oxford. So the observation here was, although the driver mutations of NPN are well understood, they're basically, color, MIPL, and of course the JAK2. Genetic alterations there's really quite a lot of morphological overlap. And pathologists essentially are required to sort of review the cases, although targeted sequencing is done to basically say which subtype, whether it's pre-myelofibrotic ET or PV, the patient has to then plan an adequate treatment. So the first thing we looked at was the shape of megakaryocytes. So you see here, these are a panel of megakaryocytes as you see them in the WHO guidelines, and they have slightly unintuitive names. It sort of starts with loose clustering, dense clustering over a Staghorn nucleus, and it's sort of accepted in the field that while this nomenclature exists, is actually not as easy to replicate in different medical centers. So what we developed at the time was a workflow for phenotyping cells. So you see here, we essentially process the entire trephine. We then use a machine learning model to detect megakaryocytes, to label them, to segment them, and then we perform clustering. Initially, our model had 9 different phenotypes, and you see them on the right. So we can superimpose them on the slide. That's what you see on the right in panel B. And then we can look at a histogram where you can now say, look, which phenotypes are really present. And our initial assumption was that this would really be the key information. For instance, is there specific meaning to a phenotype 6 or 8, or is the ratio of these phenotypes really important? And as you see in the bottom, we generated other types of spatial statistics to look at this. What turned out to be really a lot more interesting was to look at the variation in these histograms. So what we then did is we just did very sort of simple PCA on these histograms. And what you see, first of all, this is on the right in panel A, you see that the subtypes, the known subtypes, are actually fairly well defined, almost like areas within the PCA space. So you see the myelofibrotic cases on the right, where you see the near normal ones, or to the left. And what you see here on the top is also, we had then access to a couple of patients with longitudinal biopsies. So we had two patients for 2 biopsies for 1 patient. Then you see there as a second one where we had more biopsies over time. And you see on the top, we see look at 2 patients with stable disease. And what is really interesting, if you contrast this with patients with disease progression, you see here the starting points are circles and the end points are triangles. And you really see there's a progression in this, almost like what we now call disease space. And this is a concept that we now followed on, and you see that in other analyses, that we think it's a very intuitive way of basically looking at disease states and also disease progression. So in addition to megakaryocytes, we looked at fibrosis. Fibrosis is an important assessment for NPN classification. And we look at 4 subtypes of fibrosis. These are also the subtypes as they are in the WHO guidelines. What we then, of course, try to sort of recognize is that fibrosis can be very heterogeneous. For instance, here you see fibrotic hotspots, where you see also areas where there is very relatively little fibrosis. And the idea was, could we come up with a more localized measurements that could be used to aid pathologists in the assessment of fibrosis. So we developed this concept of our continuous index of fibrosis index, which learns essentially from pairwise comparisons. So what you see here, it's a ranking model where experts essentially just rank which tile has more fibrosis than another. Based on this, we learned a score between 0 and 1, and we can now basically look at fibrosis at a very localized level, so we can see hotspots of fibrosis and we can quantify those. So here's basically a more detailed example where you see the hotspots are clearly captured, but we also see areas in which there's relatively little fibrosis. So interestingly, we can do a very similar analysis what we can do for the megakaryocytes. So we can basically look at what is the whole spectrum of disease. And you see here that also MF and pre-MF are more towards the right, where we can then look again as patients over time. So here you see two patients, and the interesting patient is really the one on the right. And this is someone that has basically pre-transplantation, is the first biopsy, and then we have post-transplantation. And you see that almost like we can map the biopsies almost like in accordance to the recovery, and we also see that the fibrosis index declines over time. So this is almost like another data point that this idea of looking at disease representations could perhaps be quite powerful. So we extended this analysis to a clinical trial. Now, Ground Truth Labs conducted is a retrospective analysis of a clinical trial that Roche ran. This is a multi-center study of this Tinpentraxin alpha. It's a drug that has not progressed throughout development, but the important thing what we had access to is we had access to 50 patients with 3 time points. So we had them at screening and 2 follow-up stages. And the first thing I want to show you is this is a drug which is supposed to be an antifibrotic drug. So the entry criteria for the trial was that patients would actually have a form of myelofibrosis. But you see here that if we look at the all the bone marrow trophines with our basically fibrosis index, you see a significant variation in the data. And if we now basically plot all these cases onto almost like this disease space we talked about, we sort of see that a few patients are really well outside what the myelofibrotic spectrum we have seen before. And this was basically our first question to them. They were quite surprised. And you see also on the left, you see that the CIF score has a much higher dynamic range, for instance, than manual scoring. Now, the last piece I want to show in this context is when you now look at response to therapy, if we apply the technique I just described, we can, of course, subdivide patients in a far more detailed way. So we can, first of all, look at the overall reduction in CIF score or the overall increase in CIF score. And then we can also see that there are actually a non-negligible number of patients that do respond to the drug. And of course, if you do all of this without almost like differentiating those patient groups, you get a much different picture on how successful the trial was. So this has been published since, and it's also a nice, almost like proof point that this kind of quantitative analysis of, in this case, fibrosis, can really provide a utility in the clinic as well as for clinical trials. So what I want to explain now is how we extend this analysis with the help of spatial transcriptomics. So far, We have utilized just morphology extracted from standard H&E slides. And we've also used specific IHC markers. And the question is now, actually, can you almost like drive discovery through localized genomic analysis? And can we translate some of that back? In some sense, we have already seen CMS as an example where, for instance, the disease stratification was done based on RNA sequencing. And we were able to show that we can essentially map this back to morphology. So what we did here, with more or less samples from the 10x platform, we extended this to spatial transcriptomics. So now we have the whole slide in DAPI. We have the H&E image here. We have RNA, and then we also have the almost like analysis of neighborhoods. And again, I give a couple more details on this, but we also have a very detailed annotations where two pathologists essentially identified all the intertrabecular spaces, which they said would be healthy and preserved enough that we can analyze them. And importantly, here, the spatial transcriptomic is registered to the H&E image. So we did this on a cohort of 31 bone marrows, and these were quite carefully selected so that we start with a set of normal ones, PV, pre-PMF, ET, and myelofibrosis. So we wanted to cover the entire spectrum of disease. So initially we started out with 31. This has since been extended. So what we developed is an entire pipeline. We essentially use the 10x genomics platform. We do registration. We segment specific features we are interested in, and this goes beyond the segmentation that 10x can provide. I already mentioned that we do a detailed level of quality control. Then, of course, we do supervised and unsupervised clustering for cellular annotation. We then incorporate this with some H&E features we were interested in, and then provide an analysis of cellular neighborhoods. So which are the H&E based features we're interested in? We're, of course, interested in bone and fat. We can also use our notion of megakaryocytes phenotype, map these with the regions that are all spatially preserved, and combine this also with the measures of the fibrosis I just described previously. So for all the 31 cases, we now have a complete data set, which essentially consists out of H&E and cell labels, the sparsely preserved regions, the fibrosis scores, the notion of bone and fat, and also then the intertrabecular areas, supervised cellular neighborhoods, and then unsupervised cellular neighborhoods. And we sort of are extending this analysis now, where we sort of say, is there actually some information now in here that has been, for instance, not analyzed, or couldn't be analyzed by pathologists by sort of gross morphology. And so one of the conclusions is, for instance, that we probably see a lot more megakaryocytes than we have initially seen. So we see a lot more immature cells that, for instance, are in the bone marrow. And of course, the question is, what of these results could be translated back into a morphological analysis? So we are now almost like coming to the topic of what barriers do we see at the moment in biological imaging, especially when we're imaging in tissues. So at the top, you see almost like the what we can do in terms of H&E and spatial transcriptomics. On the bottom, you see now a slide which has been imaged using a light sheet microscope in 3D, which in this case is a prostate section, but this is something we have already established. We had a funding from Prostate Cancer UK to image prostate biopsies in 3D. But the question is almost like, what can't we do if we look at the whole 3D context of the tissue? So of course, we lack spatial context if we only look at thin slices, which you've mostly seen throughout the talk. On the other hand, in imaging in 3D is limited to several channels. We can't look at, for instance, 20 or 30 different channels in 3D, and we can't, at least not to date, do complete spatial transcriptomics in 3D. So the question is, how can we combine essentially analysis in 2D with that in 3D? Can we, for instance, look at key markers, then look at spatial distribution, and almost like do a discovery in 3D and then translate this back? So I've been working with colleagues at NIH a while on this concept, where we basically say, look, there are advantages of looking into 2D because we have established grading conventions, we have proven tools and experts, and of course, what I've just illustrated, we would like to look at cell morphology and content in 3D, and we also would have a far improved capability of detecting rare events in 3D. So the workflow we've already established, the analysis workflow, is that we can take a thick section, say for instance 100 micron section. We then generate a synthetic H&E, which can be inspected by a pathologist. We can, of course, segment the 2D slice, but then we can sort of look at the adjacent 3D section, and we can sort of see what is really the cellular morphology in that 3D neighborhood. We can analyze it, and we have also developed an integrated viewer that been basically combine 2D with 3D, and this is something we're trying to extend as we speak through a Wellcome Trust pilot in bioimaging technology. Here's sort of a little bit a preview of the work package in this. So at the moment, we are extending the work we're doing in 3D microscopy by developing 3D multiplexing protocols. So here, the idea is to develop, for instance, a 6 or an 8-plex assay that can be imaged on this open top light sheet we have, and then be combined with a robust sort of image quantification. The next step is then to sort of develop almost like a 3D atlas, where we systematically would go through basically small cohorts of samples, and you've seen these would be very much similar to the study we did in spatial transcriptomics. And for instance, from a cohort we understand very well, we could take a subset, image them in 3D, and then combine this with the spatial transcriptomic data to do sort of integration in terms of multimodalities from normal H&E, spatial transcriptomics, and then also multi-channel fluorescence. If you combine this with single cell studies, you will really see that a 3D analysis of a thick tissue can easily produce something up to about 10 or 15 million cells. So, some people have already developed foundation models that are just using single cell data. So we could basically use the 3D approach and complement almost like these insights with 3D morphology. We are by no means arguing that 3D will be used in the clinic anytime soon. So this is why we are trying to then develop inference models that probably just use a subset of the data, because what we really would like to do is to build translatable decision models that work on routine clinical data. So, as Birgit already mentioned, we were fortunate enough to be supported by the Stiftung Charité to establish a complementary set of work in Berlin. And the formal host of this program is Philipp Mergenthaler. So together with him, we sort of identified a few collaborations that pretty much follow the spirit of the studies I've presented. They all have a clear clinical motivation, and they're all driven by insights basically at the tissue scale. So for instance, together with Philip, we are applying the ultrastructure imaging to patient-derived neurons. We're trying to also establish some 3D imaging of tissues. And Jana Ilo has also started to work with us on basically applying some of the algorithms we have developed in the bone marrow into local cohorts in Berlin. So there are some snapshots of projects here. So for instance, with Werner Stenzel and Philip, there are plans to look at muscle pathology. With Wolfgang, Kübler, we would like to look at preclinical models of the lung. And there's also plans to apply this to cancer imaging with Inge Tinhofer and Ulrich Keilholz. And I've already mentioned work Jana has already started. So we're now at the stage where a postdoc in Berlin, that's Penda Alizareh, is in post, and we've just started, a few weeks ago to fill these collaborations with life. But I think the exciting thing is that this program of work will be extended, and also I'm sure there will be some links with the BIH that are really of value here. This is just a summary of the people that are in the group, what funding we have received in the past. And this concludes my talk, and then we can now start to answer questions.
Thank you very much, Jens, for this fantastic presentation. It's really amazing what you are doing there, how many different things. So as said at the beginning, please post your questions into the chat. Maybe while the people are thinking and typing in their questions, you mentioned something with regard, well, I'm not a microscopy expert, but unsupervised cellular neighborhoods determination. Can you expand a little bit more on that aspect? Do you really think you can find new neighborhoods determined by cellular environments we have not been aware of and which are important in the disease progression?
Yeah, so what we did is, for example, if you look on the right, you can sort of see after clustering, you can essentially label various different cell types in terms of spatial transcriptomics, right? So what we developed is almost like a neighborhood descriptor that looks at, for instance, I'm centered around the megakaryocytes, and now I see which types of cells are in my neighborhood, say in 20 microns, 50 microns, and 100 microns. And that information can be used for clustering. And what the one thing that's quite exciting, what we've tried is we have now looked at almost like the spectrum of these neighborhoods and sort of say which ones are predominantly seen in the normal and which are predominantly seen in for instance, MF cases. And we can now almost like go similarly, like you've seen our trajectories in disease space, we can now sort of say, okay, show me which cellular neighborhoods are more predominant in the normal tissue and which cellular neighborhoods are more predominant in a disease type. Yeah, of course, there will always be a mix. But you, for instance, see that there are cases, for instance, MF cases, where the bone marrow is still functioning, so they still basically respond to therapy. And the neighborhood analysis really brings or manages to differentiate these cases. To be honest, it would be I wouldn't do it justice to go into the biology because first of all, someone with a background in bone marrow will have to sort of shed light into this. But what we have seen is we just had a smaller paper at MICI where these almost like neighborhoods can really be used to sort of um, look at it in a more data-driven way. Does that give you a partial answer?
Yeah, so it's dominated by the transcriptomics. So you don't
It's
take
dominated
much.
by the transcriptomics. That's
Yeah,
right.
that was my question. So whether you really use the multi-modal information to also create these new neighborhoods, but it's dominated by the transcriptomics.
That's right. But we bring in the H&E features, right?
Yeah. Okay, and then Annika Neuschutz asked about the segmentation of the spatial transcriptomics, how challenging it really is using just the nuclei for segmentation, and what are your thoughts about?
Well, of course, 10x provides you with a segmentation protocol that's continuously improving. We found it does not really work well on large cells, so this is why we're combining it with the megakaryocyte analysis. And we, for instance, segment the fat separately. The case, like for instance, now the signatures of fibrosis you won't be able to extract from the 10x protocol, and this is really where by combining this together with H&E and other modalities brings an advantage because you can essentially combine all these different signals into one.
And so you find it more challenging with really big cells, yeah, because we have really problems, and we also came to the conclusion that we cannot base our cellular segmentation solely on the transcriptomics and use multiplex immunohistology to guide the segmentation, and rather struggle with some irregularly shaped cell types and some smaller immune cell types, so not the big ones necessarily.
Yeah, I think you see this sort of relates a little bit. I think there will always be performance limitations to work in thin sections, right? Because the cytoplasm in most of these algorithms is just estimated. But what I think this is where we're hoping that, for instance, if we look at cellular morphology in 3D and combining this data, really would open up some sort of new opportunities there.
Yeah. Okay, then let's move on. And so Lucas has the question: how far is this really from routine application into routine diagnostic, and how skilled do the clinicians have to be? What are your thoughts about on that?
Well, we have now, let's split this in multiple sections. So the fibrosis algorithm, we have now basically conducted a first clinical evaluation study. So the data is produced, the data is processed externally, through a company. But then we are trying to sort of see if these kinds of fibrosis maps really aid to get better concordance amongst pathologists. So we see some improvements. I mean, they are not drastic, but we see improvements by a few percentage points. And we're also now combining this with more with risk prediction. So I think this is something where, as pathologists understand what, for instance, the limitations of the technologies are, this is something that probably can be translated in the near future. The ST work, I think, in terms of throughput and cost, is it's at the moment it's still prohibitively expensive. And we also spend, or pathologists we work with spend a lot of time on quality control before processing the samples. So, for example, 10x really discouraged us from starting in bone marrow because they said other groups couldn't do it. So here I think it's really careful training from tissue, really cutting up to processing and analysis are really required. I personally don't really see spatial transcriptomic to be widely used in the clinic. Maybe in selective tests, but I think the cost is just still very high.
Yeah, but the other modalities probably are in some diseases sufficient to
Yeah.
and people work on it. I know that in many places to really move that into AI guided decision making. Probably the bone marrow is challenging due to many aspects, but yeah. So I really hope that also Germany will use that in the near future more extensively. And then Matthias has, and I really need to read out the whole question because I'm not the expert here, has written in the chat. You mentioned that 3D imaging is restricted to a couple of channels, but 2D multiplex is missing spatial context. Ultra high-plex 2D technologies are pretty high throughput by now. So could you collect a lot of 2D samples and deduce from this set 3D properties? However, I rarely see stereology being used to deduce spatial context, even though AI would be a good way to sample these 2D sections.
Yeah, so a few people are doing this. They are, we can basically take serial sections and process them through basically multiplexed imaging. There are some limitations. So serial sections are never perfect. And because the tissue can really deform quite a bit, you spend quite a bit of time in registration. Now, the registration techniques have really advanced, and it's probably possible that groups that do this. However, we really think that we want to look at the almost like full intact 3D tissue. We think there's certain advantages, especially when you get higher throughput. You can do this basically in one workflow. And then the question is also, you probably don't need that level of detail on each and every slice. So, for example, imagine you have a sandwich, you have a thin slice at the top, a 3D section, and a thin slice at the bottom. Then you, for instance, can sort of see, what are, for instance, specific cell types, or what is specific about this particular tissue. Combine essentially the 2D and the 3D information in an almost like effective multi-dimensional readout.
Yeah, that sounds really interesting. I have a question to the first part. Are you planning to move that the mitochondrial organelle imaging or analysis with the expansion microscopy? Are you going to move that into drug screening approaches also,
Well, that
and
would be
maybe human model systems or something? Because I that can add on a lot of, or give more information on top of what we now use, just soluble mediators, not really describing the cellular states that well. Is that the idea
Yeah,
behind it?
so I think in some sense Philip is pioneering this Mergenthaler in Berlin a little bit already. So he's using the patient-derived cells. So these are now well categorized. But I think, Birgit, what you said is almost like this
Yeah,
eventually opens up a new dimension for drug screening.
yeah.
And I think what we have shown now, we probably release it within the next 10 days, is um, the largest mitochondria data set that's available to date. So I think we have imaged over 15,000 mitochondria um, compared to the EM data sets that were available before. This is really quite a lot more. But of course, now the next step is, could we almost like scale this up for almost like high content screening? And I think it probably would require someone that really sees the value of that application, that has a specific question where we could sort of say, look, really, we could only address that if we look at basically ultrastructure in that detail.
Mm,
A couple of problems have been mentioned to us. For instance, people still don't understand how vaccine bind on an intracellular level, but
mm.
or there are certain, like for instance, also toxicity questions that could be resolved. But I think it would really need someone that is very familiar in this space and knows exactly, because there's some technology development that would be needed before this could be achieved.
Yeah, but I think it's really attractive, as we had our little discussion before
Yeah.
the webinar started. Immune cells change their, for instance, organelle structures and amount dramatically upon different types of activation, and one can use that or implement that into drug screenings. And then, of course, you have certain diseases caused by defects in mitochondria, and so there's a lot of potential, I would say. And just as an add-on question, you are using the mito tracker to define or the segment the mitochondrial areas, but it is dependent on the potential. Do you consider? So did you come across some problems?
Well, there are some problems. You get a lot of
Mm,
unspecific binding with the mito
yeah.
tracker. And I think you just need to recognize that so far the work have been driven forward by people that were not experts in mitochondrial
Mm,
biology. So of course, the whole labeling could be optimized. There
hmm.
would be, a lot more information one could glean from that. We have essentially just used labels that were commercially available that we had access to because we wanted to demonstrate
Yeah. Proof
in
of principle.
principle that
Yeah.
it can be scaled up.
Yeah. Okay. Fantastic. I don't see any more questions in the chat. And it was really amazing. I learned a lot. And I think my group would be more than happy to collaborate more specifically with you in the future and different aspects. So thank you very much, Jens, again. And looking forward to see you in person in Berlin in the near future.
Yes, I'm looking forward to get to know you, especially through the BIH as well. Thank you so much. And
Thanks.
see you then. Bye bye.
Bye.